All right, guys. Hi, this is Mr. London. This is my third time trying to record this video. Let's hope this one is the last time. So we've talked about the structure of DNA. We've talked about how DNA replicates. Today we're going to talk about protein synthesis, and I'll tell you what that means in a second. So again, today we're going to talk about what DNA actually does, what it makes, how it actually works. Okay, and what just happened? So every gene, okay, and a gene is a segment of DNA, codes for a different protein. So that's the main thing I want you guys to know from this whole lecture is that the whole purpose of DNA and genes is to make proteins. That's the product that DNA makes because our bodies are built of protein. Our hair is made of a protein called keratin. Our skin is held together by proteins. One of them is collagen. Our muscles are built of protein. Most of the enzymes that control the reactions in our body are protein. Proteins do other things too. They're involved in signaling and communication and transporting things around the body. So every gene makes a different protein. That's what I really want you guys to know. But proteins are made in the cytoplasm on ribosomes, right? And DNA is in the nucleus. And DNA never leaves the nucleus. It's protected in the nucleus. So how do we get that genetic information from the nucleus to the ribosome? We need a messenger, and that messenger is called messenger RNA. So that messenger RNA is also nucleic acid. So it's also carries genetic information. But it carries that genetic information from the DNA to the ribosome. That's why it's called messenger RNA, aka mRNA. All right. It's a two-step process. First, transcription happens. That happens in the nucleus because that's where DNA is. And that's the process of taking DNA and converting it into messenger RNA. The second step is translation. And that's the process of converting the messenger RNA into a protein chain, also known as a polypeptide. Okay, it's called translation because we're translating a genetic language into a protein language. So we're converting nucleotides and nucleic acids into a chain of amino acids because amino acids are the building block of proteins. So again, the whole purpose is to make proteins, and we need some way of taking that message to the ribosome. That's where mRNA comes from. So again, DNA is in the nucleus. It's converted into DNA. It's converted into messenger RNA. That messenger RNA then sneaks out of the nucleus to the ribosome, which is in the cytoplasm. Okay, the ribosome slides along the messenger RNA, making a protein chain, also known as a polypeptide. So let's talk about the difference in DNA and RNA. So what do they have in common? They're both nucleic acids. They're not carbohydrates. They're not lipids. They're nucleic acids, right? So they carry genetic information. But DNA is double-stranded. That's why it's called the double helix. RNA is single-stranded. DNA uses the four bases A, T, C, G, adenine, thymine, cytosine, and guanine. RNA uses A, U, C, G, adenine, uracil, cytosine, and guanine. Okay, and the base pairing rules are a little different too. With RNA, A always binds to U, and C still binds to G. That hasn't changed. But with RNA, everywhere you want to put a T, it's a U instead. Okay, guys? The third difference is the sugar that these two nucleic acids use. DNA uses deoxyribose. That's why it's called deoxyribonucleic acid. RNA uses ribose, which is why it's called ribonucleic acid. Finally, there's many types of RNA. We already talked about one, messenger RNA. There's only one type of DNA. Okay, let's move on. So again, the three types of RNA, there's mRNA, which carries the message from the DNA to the ribosome. Then there's tRNA, which is kind of like a taxi cab. We're going to talk more in depth later, but it basically is shuttles amino acids to the ribosome to help build that protein. Okay, so each tRNA drops off a different amino acid because proteins are built of amino acids. Okay, and the third kind is rRNA, also known as ribosomal RNA. And it basically is the same thing as a ribosome because ribosomes are made of RNA. All right, so again, we've already discussed that, but is another image. The top one is the ribosome, that orange thing sliding along the mRNA. That's a ribosome, aka rRNA. We've got our blue line here. That's messenger RNA. And then these green things, they're transfer RNA. So each one drops off an amino acids which are then linked together to form a protein chain. Okay, so transcription again is that first step in the process of making a protein. We're making a messenger RNA from DNA. Okay, there's one enzyme that you should know. It's called RNA polymerase. RNA polymerase has two jobs. It unzips the DNA and it makes RNA. Okay, just like DNA polymerase makes DNA, RNA polymerase makes RNA. Okay, so it makes RNA. So how does transcription actually work? The RNA polymerase, which is not shown here, unzips the DNA, opening up the DNA, just like we have here, kind of like DNA replication. Okay, so it opens up the DNA, which is our double-stranded thing here. So the DNA is shown in kind of that grayish blue. And then our new mRNA strand is made, which is complementary to one of the DNA strands. So that's that yellowish line. That's our messenger RNA. Again, it's single-stranded. And it's getting made because the messenger RNA is complementary to our DNA strand. I like this picture better because it actually shows the RNA polymerase. So notice how that green thing, the RNA polymerase, is sliding along our double helix, unzipping it while also gluing on our little bases, our RNA bases, to that DNA. So the DNA is the blue one. The green thing is the RNA polymerase, an enzyme that is making the red messenger RNA. Notice the base pairing here. Everywhere you have a G, you have an RNA-C. Everywhere you have a T, you have an RNA-A. But you never have a T now. Every time you want to add a T, you're adding a U because RNA uses U instead of T. So the way I help my students remember, it's ants underground, cars in the garage, because A is complementary to U and C is complementary to G. Okay? So where does transcription happen? It starts in the nucleus. Once that messenger RNA is finished being made, transcription is finished. Okay, but that messenger RNA has to leave the nucleus. So it leaves the nucleus through these tiny holes in the nuclear envelope, which is the membrane around the nucleus, and then it makes its way to a ribosome, which can be either attached to the rough ER or just floating around in the cytoplasm. So this is a really cool image of RNA polymerase. Is the purple thing. It's sliding along our DNA, which is that blue double helix, sliding along the DNA, making messenger RNA, which is represented by this red line right here, right, coming out of the RNA polymerase. Okay. So again, when we zoom in, we've got our RNA polymerase unzipping the DNA, adding bases, adding RNA bases. So those red things that are floating around are RNA bases getting added, constructing the messenger RNA. So how does this work? So it starts with DNA, which is double-stranded, right? So the RNA polymerase unzips it, and then this is when the magic happens. So now we're going to transcribe this DNA into RNA. Isn't that exciting, guys? So what are we going to do? How are we going to do it? Remember, ants, underground, cars in the garage. But RNA still uses A. So T still goes with A. But for the second letter, we're on the bottom. We're transcribing the bottom strand of DNA. Instead of putting a T with that A, like we did with DNA replication, we're going to put a U. So it's going to be A, U, G. That hasn't changed. C is still with G, and G with C. So it's going to be A, U, G, C, C, U, A, G. All right. So ants, underground, cars in the garage. Okay. So A is complementary to U, C is complementary to G, and vice versa. All right, guys, step 2, translation. All right, let the fun begin. Now we're going to start making a protein. So how does that actually happen? This happens at the ribosome. So how do we translate this genetic code? We translate it 3 letters at a time. So every 3 letters on our mRNA, every 3 bases, I shouldn't say letters, they're bases, is a different codon. And each codon codes for a different amino acid or codes for an amino acid, okay? So every 3 bases codes for a different amino acid. And the tRNA, which brings the amino acids, has a complementary sequence called an anticodon. So the tRNA, the amino acid that the tRNA brings is dependent on the codon. So on the bases, I'll show you guys what that means. So here we've got 3 bases on our messenger RNA. That's called a codon. So GCA codes for a specific amino acid. Okay, so a complementary tRNA with the anticodon that's CGU will attach that codon and bring the appropriate amino acid that is determined by that codon. And how does this is just a table to help you learn, help you figure out which codon goes with which amino acid. So there are 20 different amino acids, and these are the abbreviations. So LEU is an amino acid, leucine. TYR up here is tyrosine. ALA is alanine. So these are all different amino acids. And the 3 letters to the left of those amino acids are codons. So it's the codon on the messenger RNA that determines which amino acid is going to be brought to the ribosome, okay, by that tRNA. So let's practice. Let's just look at this image of how it works. So this red line represents our messenger RNA. This brown thing is the ribosome. This blue squiggly line represents tRNA. So the tRNA has an anticodon on it, which is that blue, purple, yellow thing protruding from the bottom of it. That's the anticodon. It matches up with that's the anticodon. It matches up with the codon on the messenger RNA, attaches to it, and brings the amino acid that matches up with that codon as well. All right, guys. So let's practice. All right, so we've got a messenger RNA sequence here. All right, so let's figure out what amino acids are going to be made from this gene. All right, so our first codon is GCU. So we're going to go to our chart. We're going to go to our conversion chart, GCU. So the first letter is G. So we're going to go here on the left side, and that's G. And for our second letter, we're going to look at the top, and we've got C right here. So where does the G and the C intersect? It's this bottom row, this second row on the bottom, okay? So GCU is right here at the top, right here, and that codes for the amino acid alanine, Okay, so that codon makes alanine. So I want you guys to pause the video and try to use the conversion chart that you have in front of you and figure out what the next 6 amino acids are going to be based on these codons 2 through 7. All right, guys, so now you've hit play again. You're ready to check your answers. You should have gotten, and if you haven't done it, pause it again. And when you're done, hit play again. All right, so we've got another, this is what you should have gotten. All right, glutamic acid, leucine, arginine, serine, and then you've got a stop codon. So every gene ends with a stop codon. That basically tells the messenger RNA that it's finished. So the messenger RNA then detaches from the ribosome. The protein chain detaches from the tRNA, and it goes on its way. All right. So every gene starts, and every protein starts with a start codon. That tells the tRNA and the ribosome to start making a protein, and it always ends with a stop codon. So let's look at the. I'm going to have you guys label an image. So I'm going to have you practice by looking at this diagram right here, showing the process of translation. So the ribosome is shown by this like big bubble thing. It's got actually 2 parts, one below the messenger RNA and one on the top of the messenger RNA. It kind of clamps down on the messenger RNA and slides along it, reading that genetic sequence as it assembles the protein chain. So that's the ribosome. That's our messenger RNA. And our tRNAs then drop off their amino acids. They attach their amino acids to the chain that already exists, and they leave. Okay, until that protein chain hits a stop codon, and then that protein detaches and goes and gets shuttled to wherever it needs to go. Okay, it usually goes to the Golgi apparatus to be packaged, modified, and shipped off. Remember, the Golgi apparatus is like the post office of the cell. So again, guys, tRNA is the thing that drops off the amino acids. So it basically attaches to a specific codon. So UUA represents the anticodon. And notice how it's complementary to our codon AAU. Okay? So a lot of vocabulary here, but we're going to go over it again and again. All right, guys. So again, every gene starts with a start codon. I love this. This is an animated pic. This is transcription. We're making our messenger RNA. And now we're doing translation. So our messenger RNA then attaches to the ribosome. The ribosome slides along that messenger RNA, reading that genetic code, assembling a protein chain with the help of our friend tRNA, which drops off amino acids. Okay, so one at a time, they drop off amino acids. Those amino acids link together to form a protein chain, also known as a polypeptide, because they're linked by peptide bonds. So what is this? That's the messenger RNA, so that's the codon. Notice how it's complementary to the anticodon on the tRNA. Okay, but it's the codon that determines which amino acid is the tRNA brings. Okay, and that bottom thing represents our protein chain. So each of these circles is a different amino acid: methionine, serine, leucine, lysine, arginine. All right, so again, this purple thing is our ribosome. Cat is scratching at the door. This is our messenger RNA, and that's our tRNA dropping off the gray things, which are amino acids. All right, guys, we're going to stop there. So if you guys have any questions, just email me. Feel free to watch this video again if I went too fast.